Hello everyone, my name is Aram Khoshnaw. I'm a biologist from Iraqi Kurdistan. In this presentation, I'm going to explain my research experience at the Young Scientist Program. In this program, I chose to work under the supervision of Professor Shiladita Dasarma and Priya Dasarma. And the title of our project was regarding the evolution and survival of ancient microbes using a bioinformatics approach. The ancient microbes that we used in our project were called halophiles. These halophiles could also be called halophilic archaea. These are archaea or microorganisms that love salt-rich environments, such as brines and salt lakes. As we can see from the figure next to it, for example, the Dead Sea has a high concentration of salt, and halophilic archaea have been isolated from these environments. The abilities of these microbes in survival into these harsh environments leads to research questions and interests regarding the evolution of life and molecules, using them as models for astrobiology research and biotechnological applications. Furthermore, these microbes are hypothesized to survive the environments of Mars because they can resist to very harsh environments such as high salt concentration and radiation and living next to toxic metals. In my project, I tried to correlate DNA replication gene histories to the evolutionary history of haloarchaea. In my project, I used bioinformatics tools to investigate DNA replication evolution in haloarchaea. I practiced basic bioinformatics tools such as creating a local blastable database of haloarchaeal genomes and then blasting sequences of interest to these local databases and understanding and evaluating blast results and what they mean, which is an important skill for all of us, and then retrieving sequences from MCBI databases for further analysis and comparison. These were essential steps before performing more important skills in evolutionary relationship between sequences, such as multiple sequence alignment and phylogenetic trees of these genomes or proteomes. Then we try to find similarity between gene sequences. For that, we use crystal omega to align multiple sequences and to look at similarity and person identity between them. Then we try to use these alignments to create heat maps of relationship between the protein sequences that we had. And these were essential steps to do before going to the phylogenetic tree reconstructions. For example, as we can see from these two figures, in the figure 1, which is labeled as A, I have aligned some sequences. From there, I can see from the colors and also from eye investigations, I can see that there are some similarities in which part they are aligned well, in which they are not, and in the aligned part, what are the similarities and the differences between the sequences. And also we have the heat map next to it, which shows the relationship and the percent of similarity between the sequences, which can guide you to further steps in the evolutionary investigation. Then we try to explore evolutionary relationship between the protein and gene sequences. In yet another big step, we learned how to create phylogenetic trees. For example, creating a 16S trees of haloarchaea members, and then creating trees for protein sequences that we had, and then investigate these protein sequences with the tree of life and also with the trees that we had for the taxonomy of haloarchaea. From there, deducting and making conclusions regarding the evolutionary processes and interpretations regarding these important microorganisms that can be used for a lot of interesting research questions. Thank you for listening.